A lot of the people I want to thank are not in the room yet, so they'll have to watch this on video. But, you know, this conference was put together with, as you heard yesterday, with the Farmworker Association of Florida, and Tirsa Moreno and Jeannie Economos played an incredibly important role in bringing us all together here. And I wanted, you met Randy Abate last night, law professor at the Florida A&M School of Law, who is an incredible host, and if he were here, I'd ask him to stand up and so you could all acknowledge him. Then Jennifer Taylor with the statewide small farms program at FAMU up in Tallahassee, also a great contributor to this event. Another, you know, she's at the main branch at Florida AMU. The only guy of this group that's here is Marty, so he can stand up. And we really want to thank you, Marty, for your contribution in helping to pull this all together. Yesterday on the Toxic Tour, we visited communities and heard stories of farm workers, the working and living conditions, the incredibly hard work, the poor wages, the persistent pesticide exposure, and the fear that raising issues of concern would cost workers their job. The contamination of Lake Apopka and the workers who work the poisoned farmland live today in communities that are contaminated by incinerators and dump sites and Superfund sites. We heard about the work of the Farmworker Association of Florida as the organization works to turn frustration into empowerment. Train workers, advocate for changes in policies and practices, advocate for a living wage, and protection from toxic chemical exposure. We then visited Uncle Matt's organic orange groves. There we met stewards of the land who had adopted practices for soil fertility with the mantra on the farm that the minerals drive the bus. And we'll hear more about the minerals driving the bus in some of the workshops today. The introduction of beneficial insects, parasitoids, and we learned about the success of their practices in managing of the Asian citrus psyllid, which transmits the citrus greening bacterium, I guess. The results are as good or better than the chemical-intensive approach, which recommends soil-applied systemic insecticides, imidacloprid, thiamethoxam, and clothianidin. The juxtaposition of these two experiences on the Toxic Tour, the juxtaposition of these two really encapsulates what we're doing here in this conference, addressing both the challenges and the promises that we face. And the reason we're here is to confront those challenges and promises. Knowing of the exploitation of farm workers, we as a community must not forget that we cannot discuss sustainability without addressing the complete context in which we grow food. A large industrial-scale farming operation cannot be sustainable if it is utilizing chemical inputs that adversely affect ecological systems. And these ecological systems in which these agricultural operations are functioning. At the same time, an organic operation cannot be sustainable while addressing source issues of biodiversity, ecological balance, and the soil food web if the workers in the operation are not treated humanely and paid a living wage. That's the message I took away from yesterday. I hope you did as well. We must reconcile these two things. If we apply a key core principle and value of food production systems, and I don't know if you all can guess where this value comes from, but it goes like this: optimize the health and productivity of interdependent communities of soil life, plants, animals, and people. That comes from a 1995 document produced by the National Organic Standards Board. Optimize the health and productivity of interdependent communities of soil life, plants, animals, and people. And that's what we'll be talking about today. So while the Organic Foods Production Act may not be as explicit as we would like in embracing social justice, it is clear that no system of food production is healthy unless the people working in that system are healthy. We don't typically associate poverty or modern-day slavery as a condition that is healthy for those impoverished or enslaved. It's another message that we got yesterday. The food production and delivery system is complex, and there are a lot of institutional pressures that affect the resources available to growers and consumers. However, while the system is complex, the values of humane treatment of workers, a safe workplace, living wage are simple bottom lines, and that must be integral to our work. Management of non-agricultural land and buildings, which we'll also talk about at this conference, also involves complexities of practices, policies, design, maintenance, and other institutional barriers to change. We hold this conference to address the complexities in a way that enables us to be effective advocates for change. The science in toxicology is complex, the policies are complex, the law is complex. Rather than disempowering advocates with the complexities that seem like barriers to change, we spend time breaking down those complexities so that we as change agents are empowered to cut through them to bring us back to the fundamental and foundational principles and values to protect the fragile web of life. The complexities must never be a reason not to act or be a barrier to change as we move forward with our vision. Now, on a personal note here, I myself, as a young person, traveled to Apopka many times in the late 70s, starting in 1978, working for a social change organization, and in collaboration with Farmworker organizations. We held a series of forums. That's how this meeting got its name, in Ocoee, Florida, which is outside Apopka. And we did the same thing in the Rio Grande Valley, in McAllen, Texas, and in Salinas Valley in Salinas, California. My charge, with the support of an EPA grant at that time, which you'll all appreciate, was to create the record to support a federal worker protection standard. That's the late 70s. At that time, the only worker protection at the national level was a half-page in the Code of Federal Regulations that said workers shall not re-enter the fields until the sprays have dried and the dusts have settled. With the help of the National Farmworker Ministry and the sisters there, and the staff to the Florida Rural Legal Services and the Migrant Health Clinic, we visited with farm workers in the labor camps that Jeannie talked about yesterday, who told us stories, their stories of poisoning and illness, along the same lines of the photographs, those beautiful photographs you can see outside here. And we heard the stories of poisonings and illnesses. In addition to farm workers, we brought family farmers to share their stories of pesticide poisonings, which were strikingly similar. Small farmers' experiences were strikingly similar to the farm workers. We brought federal officials appointed by the president to listen to these stories, and over a decade after that work, a worker protection standard was adopted, incremental change at best. It did not take too long to realize that unless we work to change agriculture's reliance on toxic chemicals, the attempts at, as EPA says, mitigating risks with personal protective equipment, re-entry standards, hand washing and sanitation facilities, clean drinking water, prohibiting children in the field, posting a notification, training, helped but did not address the underlying dependency on toxic chemical use that created an inherently unsafe workplace and contaminated nearby communities, waterways, and the larger environment. We learned quickly that big agriculture and chemical companies, as we heard yesterday, would not stand by, I learned firsthand, and allow EPA to fund a bunch of change agents to, as they called it, destroy American agriculture. And that's how they described our work. While continuing to deliver the message of increased protection for workers, we started focusing on advancing efforts to grow a fledgling organic sector. And that was characterized at the time as hippies and backyard gardeners like Marty, whose farming strategies could never work on a commercial scale. We took the issue to Congress, and we introduced the bipartisan Can you imagine that, a bipartisan bill? The Organic Farming Act in 1982, and then changed its name to the Agricultural Productivity Act, which made more sense to Congress, which passed in 1985 and set up the Low Input Sustainable Agriculture Program at USDA, which today is the SARE program, which many of you know about as the Sustainable Ag Research and Education Program. Now that program, as organic, is contrary to the culture of the agency that houses it, because that agency, USDA, has a history of supporting agribusiness, fence-row to fence-row agriculture, and these programs supported research on organic production, which we're trying to institutionalize within the USDA. Then, as organic grew with numerous voluntary and state standards, flash forward to 1990 and the passage of the Organic Foods Production Act, which gave new credibility to organic, spurred on by public outrage, consumer outrage. This is field to table. We're now at the table where the public did not want carcinogens in its food. And the ALAR, you recall ALAR, which I can pronounce this, unsymmetrical dimethylhydrazine, was found in our food. We are making choices every day and have dramatic generational consequences in terms of protection of health and the environment. And we have choices. And this is hopefully why we're here, because we want to be more effective at making the right choice and being advocates for the right choice. Do we choose approaches that ignore the health of honeybees and pollinators, or choose a path that is protective of sensitive species that are integral to the web of life? Do we embrace risk assessment approaches to managing toxic chemicals that ignore the most vulnerable among us, many of whom have pre-existing conditions, and leave key effects out, like endocrine disruption, which we'll be hearing about from Lou Guillette and Tyrone Hayes, untested, or establish precautionary approaches and policies that seek to protect those who are most threatened? Do we choose approaches that fundamentally manipulate nature and life through genetically engineered plants, or do we advance practices that are in sync with our ecology and natural systems? Do we advance methods that ignore the social and economic impact on people and communities, or choose practices and approaches that are socially just? The neonicotinoids, which we heard about last night, associated with severe pollinator decline. If you just look at that as an example, you can see EPA registering a toxic chemical without all the answers that it needs, requiring studies that are deemed unacceptable or deemed missing over a 7-year period, without telling the public that the chemical is conditionally registered, as it did in February of 2011. Who should have the burden of proof? And why don't we err on the side of precaution? Meanwhile, as bees disappear in extraordinary numbers and bee colonies collapse, EPA says it doesn't know exactly why. The other name that's come up in this conference is Rachel Carson. Her book, as we've heard, Silent Spring, is really a clarion call for caution. I really urge you to read it, Marty, very carefully. Written during a then emerging chemical age of the 1950s. Ms. Carson's book introduced science-based understanding, and this is her quote: "The most alarming of all man's assaults upon the environment," in the 1960s she wrote this, "assaults upon the environment is the contamination of air, earth, rivers, and sea with dangerous and even lethal materials." The book catalogs the severe problems associated with our societal embrace of DDT at the time, but also other chlorinated hydrocarbons, organophosphates, carbamates, chemicals we're still struggling with, presumed to be effective and protective tools in managing unwanted insects and vegetation in agriculture, gardens, and homes. We keep adding chemicals to this list with escalating harm. We now add the neonicotinoids to that list. Incredibly, highly toxic, persistent, and now systemic chemicals. Ms. Carson, a marine biologist, science writer, poet, author, warned us to respect nature, support it, harness the benefits it offers to us, and never think that we can overpower it. The real-world examples that she provides in Silent Springs are stunning, from impacts on salmon, aquatic organisms, birds, beneficial insects, to humans. She warns us about pesticide uses related to rangeland, utilities, brush control, forestry, agriculture, private lawns, parks, and golf courses. So Rachel Carson really is asking us to think holistically. She says, is it reasonable to suppose that we can apply broad-spectrum insecticides to kill the burrowing larval stages of a crop-destroying insect, for example, without also killing the good insects whose function may be essential? We heard about this yesterday at Uncle Matt's. So while we may focus on a bad actor chemical like DDT or the neonicotinoids, in our campaign to protect pollinators, yes, we do focus on neonicotinoids. Systemic pesticides, as I said, that contaminates the whole plant. But we're also addressing the failure to address the complexities that contribute to colony collapse disorder. EPA's failure to regulate these chemicals is symptomatic, and when we talk about its failure, we're going to hear specific chemicals cited over the course of this conference. But those failures to regulate these chemicals is really symptomatic of the agency's failure to regulate in the context of biological systems, and that's the key here. In effect, the complexities of nature are virtually ignored by the regulatory review. One incredibly bright spot in the spirit of Rachel Carson is our federal organic law that requires us to ask these very questions, whole systems questions, in regulating certified organic systems and materials. Rachel Carson's vision, her core beliefs, her scientific assessment, her analysis of the value of preventive solutions implore us to take the holistic and organic steps necessary to save bees and people, workers and consumers. And here's my final thought. It is because of the importance of the organic law that we will ask and continue to ask the community that has struggled to achieve its passage and its holistic orientation, that we fight to protect its integrity. So we are building public trust. That's our goal. One of our goals, I think, when we leave here is to answer the question, how do we build public trust in a system that is accountable to those who believe that their decisions on food purchasing are reflected in a trusted decision-making process with public input, scientific assessment, and assessment of the viability of practices on the farm. So that's your charge. That's the charge of this conference. We hope that this conference really informs what you do when you leave here, that this is not a static event, that is something that doesn't support your continued work. I know you all in this room are committed to the work of this conference, and we at Beyond Pesticides, and I know the other organizations that are here and on the program, are committed to working with you as you work in your communities, at the community level, and engage in the process of change. Change for the present and change for future generations. So with that, too long an introduction, I turn this over to Terry Shistar, our longest standing board member, still standing, at Beyond Pesticides, a PhD ecologist, University of Kansas, from Lawrence, Kansas, and Fillmore, or Nottoway, we call it, Missouri. So thanks, Ted.
Hi. As many of you know, every year at the Beyond Pesticides Forum, we try to bring cutting-edge science to the forum, and the field of endocrine disruption has been a particular focus of ours. When Lou Guillette first spoke at the forum, he was introduced as the alligator penis man in honor of his research. He's one of the forum attendees' favorite speakers. Lou is considered a pioneer researcher on endocrine disruption and reproductive effects. He studied the decline of Lake Apopka's alligators, and he's currently the director of the Marine Biomedicine and Environmental Sciences Center, and Professor of Obstetrics and Gynecology at the Medical University of South Carolina, Professor at the Howard Hughes Medical Institute, and Smart State Endowed Chair in Marine Genomics at South Carolina Center of Economic Excellence. His current work examines the effect of environmental pollutants on the genetic endocrine signaling system associated with the development and functioning of the genitalia and gonads of various vertebrates. Please join me in welcoming Lou Guillette.
Well, since part of the title is alligators, I figured I had to start with an alligator picture. So, I know you saw lots of farmland yesterday. Hope you saw an alligator. Did you see an alligator?
Yes, on the way.
Oh, Jeannie, no alligators? Oh my gosh. Okay, well, we have a couple of pictures of alligators today. The title of this talk that I sent was Sentinel Species. What are they telling us? The subtitle is basically 25 years of environmental endocrine disruption research and some stories and maybe some things that I've learned. It's always kind of weird for me to give talks to groups like this because I'm talking to the choir. I don't actually have to convince you guys that the environment's important and that health of the environment's important. But what I'd like to do is to break the talk this morning into two sections. I'd like to do a little philosophical thing to talk about, and it's astonishing. Jay and I did not talk before this, so he has no idea what I was actually going to say today. But many of the points that he made, I'm going to focus on. And then I'll tell you a little bit about Lake Apopka. And I still work on Lake Apopka. I was at the University of Florida for 25 years as a professor. We still come down every year. In fact, I will be back down here in about 6 or 8 weeks. Working on Lake Apopka, working on lakes still here in Florida. And it's sad to say, but we're still finding many of the same things that we actually found 25 years ago. So with that as a thing, I thank Jay, I thank the organization. I'm very, very disappointed that they didn't have my replacement T-shirt that they gave me 14 years ago with a very nice dragonfly on it, but he tells me they're going to do it again. So. See, that, I think you have it. I was almost going to wear that. No, I have one. I still have that T-shirt. My wife keeps telling me I need to put it away or paint with it or do something, but it's one of my favorite T-shirts, so there. But I thank you all for the invitation. I thank you all for coming here on a Saturday morning. I wanted to put this up because we all kind of know it, but we don't know it, because the public relations is telling us something very different. This is actually looking at what you're in fact seeing is that this is looking at prostate, breast cancer, testicular cancer over time. And what you can see is that every single one of these is going up. What we're hearing is cancer deaths are down. That's true, thank God. But the fact is that the rate of cancer, or the increase in cancer by population, continues to increase, especially in endocrine cancers, especially in neurological cancers. Now, the premise of our understanding of disease for much of the last 50, 60, 70 years has been associated with what I call genes and germs. The fact is we think of disease either as pathogenic organisms or genetic determinism. We have a mutation we inherit from mom or dad, that leads to disease. You're exposed to ionizing radiation, leads to disease. Exposed to a couple of chemicals, leads to disease. The fact is what we now know with the Human Genome Project is that there is not going to be a gene for Alzheimer's. There's not going to be a gene for attention deficit syndrome. And in fact, most of the diseases we're dealing with, breast cancer, prostate cancer, testicular cancer, and a whole series of other things, only about 15 or 20% of the cases are associated with specific gene mutations. We are in a new world, and that new world is what is in fact epigenetic determination. That is where the environment is playing a much, much bigger role than we ever dreamed in leading to disease. And in fact, many of these diseases are not associated with specific genetic mutations. They're associated with changes in gene expression. How is your genome actually being regulated? How does that lead to a predisposition for disease? How does that in fact lead to disease? So what I'd like to do this morning, very quickly, is to kind of give you what I would maybe say is my top 5 lessons that I've actually learned in basically 25 years of doing environmental science, and then talk about alligators. And the first lesson is kids are different. Kids, embryos, whatever you want to call it, they are not little adults, and they're not big mice. They are fundamentally different in how they process chemicals and how, in fact, they grow and develop. they are in fact different, although most of the toxicology that is still done is actually done on adult animals, and then we try and predict what goes on in all the other stages of life. Let me show you one data set, which for me is stunning. And this brings this message home. This is actually DDT association with breast cancer. What you actually see on the bottom line is the age of this girl or girls. If you are less than 4 years old and you are exposed to the highest levels of environmental DDT, these are not little girls working in DDT factories, the odds ratio of developing breast cancer is astronomical. Having basically an 11-fold increase in your chances of getting the disease is astronomical from a medical perspective. In contrast, if you've reached puberty and you're greater than 14 years of age, your odds ratios of developing breast cancer are the same as if you're not exposed. So what does that actually tell us? Well, what it tells us is something which has actually been emerging and is now quite extensively funded by the National Institutes of Health, is something we call the embryonic or fetal origins of adult disease. That is, many of the diseases that we are in fact seeing today are set up during embryonic development or early childhood life. This is not a hypothesis anymore. It originally started off as something called the Barker hypothesis. It originally started off by looking at low-weight babies born from 1945 during basically what was in fact World War II, the Holland famine, which was created when the Nazis blocked off Holland, and there was a major famine in Holland. Women were pregnant during the first trimester of that famine. Their babies actually had a higher chance of developing cardiovascular disease. It was completely opposite to what was predicted. A low-weight baby later in life developing heart disease just didn't make sense originally until the developmental biologist got involved and said, well, you do know when the heart and circulatory system develops, right? During the first trimester. That's when, in fact, a lot of this set up. Well, what about if they actually went through the same low nutrition, but during the last trimester? The last trimester is when your brain is not just growing, but all those neurological systems are being set up. And so therefore, if you actually see low nutrition during late trimester, what you in fact have is a much higher incidence of depression, diabetes, schizophrenia, antisocial behaviors. So the take home is, we know that the embryo is in fact sensing the environment. This happens to be some basic things, but what about my world? My world is reproductive biology. So the Johnson Family Foundation gave me a grant a number of years ago. We brought together what were about 10 or 15 of the top people in the world in reproductive biology and developmental medicine. We looked at thousands of papers, and what we were able to show is that these 8 general diseases, or if you will, conditions we want to call them, that young women appear in a gynecology or an obstetrics practice. And what we were able to show is that every single one of these conditions, there is evidence that they can in fact be established during embryonic development. Now these are things like polycystic ovary syndrome, endometriosis, fibroids, which are epidemic in the United States, they're epidemic in Western Europe, and they continue to grow. Now yes, several of these conditions can be established by certain kinds of exposures, stress, etc. later in life during the reproductive years, but all of them have the potential basis to be set up during embryonic development. So, like in photography, timing is almost everything. And it's really critical for us to actually start thinking about when these exposures are actually taking place. Now, you heard about ecosystems, and I will hammer home today that mixtures are, in fact, the world that we're dealing with. We are never, ever, from my perspective, going to predict what goes on in a real population by studying one chemical at a time, or even studying one variable at a time. When we actually look at the health of a baby, we now know that it's not just the genetics that you inherit from mom or dad. We've always known that nutrition played a role. That's why we, in fact, propose folic acid and various other things, vitamins during pregnancy. We know that these variables are important. We know that drugs have the potential to alter the phenotype or the health of that offspring, both illegal as well as legal drugs. But what about personal care products? Well, we now know that's definitive. And we've just, in fact, confirmed it ourselves. We've just finished a 400 pregnant mom study in the last couple of months. We're basically finalizing all of the not all of the genetics, we're doing that too, but all of the statistics. And what we're actually finding, and for the first time, we actually have 200 African-American women as well as 200 Caucasian women, and we are finding that personal care products have measurable effects on their offspring. And for the first time, not just baby boys, but baby girls as well. And I'm not going to go into that data today. As I said, we're still trying to finalize the stats. But the take-home is these variables are real. More importantly, all of these phenomenon contribute to this developing offspring. So just studying contaminants in mom, or just studying genetics, or just studying personal care products is not going to give us what we need to understand about the potential health of that child later in life. Now, that doesn't mean that this group and your focus isn't important. Understand what I'm trying to say is that it is important, but it's in context to all the other things that, in fact, a young woman or a family is exposed to. And it's not just us. The fact is that every organism is dealing with the same mix. They may not be getting illegal drugs. They probably are getting illegal pesticides or whatever else into their environment. But the take-home is my alligators did not go into a physician and get a pharmaceutic. They're dealing with what we're dumping into the environment. They're basically dealing with environmental change. Oh, climate change. I'm not allowed to say that because I'm from South Carolina. But modifications in environment. So one of the things that we've actually spent a lot of time in the last 5 years trying to figure out is how do you study complexities? And so I put this up as just a very quick thing. We published a major paper in environmental science technology. It took us forever to get it through because the reviewers didn't understand the statistics. What we in fact took was a page from national security. What we in fact did is machine learning. The ability to take complex patterns like you do when you walk through the airport and they try and recognize who you are by taking your picture and running it through a database of 1 billion people. The fact is, we said, what happens if you take hundreds and hundreds of data points and put it in, can you recognize disease patterns? And so we did something a bit different. I'm at a marine lab. We're interested in algal blooms and algal toxins, largely associated with nutrients that we're dumping into the environment. We looked at 3 algal toxins, but what we actually asked is, what about metals in the environment? Because we know iron plays a role. What about temperature? And not to go into belabor the point, we basically looked at 18 different variables at 10 different levels. That's 10 to the 8, sorry, that's 18 to the 10th. We're talking millions of possible combinations. The whole NIH budget cannot do that experiment, okay? And in fact, everybody in this room, if they worked their whole careers, couldn't do that experiment. But we asked the computer, tell us 200 possible combinations randomly throughout the whole hyposphere. And we did it. And it said, you need to do a few more. So we did a total of 260 possible combinations compared to 18 to the 10th power. And what we, in fact, were able to show is that we could predict with 90% assurance what, in fact, the most complex and most important interactions were. Is 90% okay for public health? I think so. Now, is this 95% confidence in the mechanisms? No, but you know what we're now doing? We're taking those most complex interactions and we're doing the next set of experiments. So it allows us not only to test, but it gives us the next generation of hypotheses that we can actually go and ask, what are the most important combinations? And as you see, these complex relationships, these 3-dimensional relationships, are not linear dose-response curves. Lesson number 3, it's not just toxicity. We always love to think about chemicals being tested for their toxicity. Yes, chemicals kill things, but most of us are not, in fact, dealing with death. What we're dealing with is changes in health. And so it's really important that we look at different endpoints, and of course, that's partly the story of endocrine disruption. We lost 2 giants in the last couple of years in this field. The older gentleman that's sitting next to Tayaguchi in that picture is Howard Byrne. He's the guy who discovered the DES daughter, who many of us know about the DES daughter complex. Howard was my mentor, and actually Tyrone Hayes' mentor. So you have 2 Howard Byrne prodigies here talking to you today. And the woman sitting next to Howard is Mom. That's Theo Colbert. And so Theo was, in fact, the one that stimulated most of us, in fact, I would say almost all of us, to actually start thinking about endocrine disruption and being part of this. And for many years, she was the only one that actually believed that what we were doing was important. And unfortunately, I talked to her just a couple of days before she passed away here last December. The take-home is that this is no longer the endocrine disruptor hypothesis. We are absolutely sure that this is real. And it's not just real in wildlife, it's real in humans. So real that the NIH now, in fact, posts things like this, where they can actually say there's over 1,000 chemicals we now know are endocrine disruptors. And they, in fact, bridge the gamut of the kinds of things that we're exposed to every day. Endocrine disruption, it's important to recognize, is not just the ability of a chemical to bind, let's say, a hormone receptor. It's the ability for it to modify the central nervous system, the hypothalamus and the pituitary. It's the ability to actually alter, for example, how the gonads, ovaries, and testes, or thyroid, makes critical hormones that actually are involved not just in development, but in reproduction and in general health. It also modifies the liver. So in the last 25 years, my lab has actually been establishing, or has established, that all of these levels of organization can be disrupted by exposure to mixtures of agricultural chemicals, i.e. pesticides. But there's a tremendous problem. And the problem is that we don't, in fact, communicate very well with one another. And when I mean that, I mean within science. So we can understand what this guy's telling you. Anybody who's ever actually been in Africa, and we actually have a major project there, and you walk along the river, when he communicates, there's not a question what he's telling me. It's like, get out, okay? This is my territory. But the fact is, we are still trying to deal with the toxicology world and their assumptions. The assumptions, and they truly are assumptions, that are fundamentally different than many of the other fields. This was actually from a plenary I gave to the Society of Endocrinology and a couple of thousand physicians a few years ago. And all I was trying to point out is that in the field of endocrinology, for 100 years, we have known that there are things other than linear dose response curves. Inverted U-shaped dose response curves are absolutely normal in the field of endocrinology and neurobiology. But we're still battling with the EPA and regulations about how you, in fact, test chemicals that don't respond in a linear fashion. And as you can see, there's a whole bunch of other assumptions that are made, and I'd be glad to talk about these later, but that's not the basis of the talk today. Lesson number 4 is that it really is about evolutions, and evolution works on populations. The fact is, pesticides do too. Why do I bring this up? Because for some reason, when you talk about a non-lethal response, we always hear, well, I did X for 25 years, and look at me, I'm fine. Okay, and you can put X, you can put in anything you want. The fact is, there's always going to be people in a population that sprayed DDT for 1,000 years, and they're perfectly okay. It's populations. Populations have a range of effects. And one of the central problems that we actually have is that most of the regulatory work that's being done is done on model species. What's wrong with model species? They're model species. They literally have been developed so there's no genetic diversity, or very little. So if you're asking about real populations, if you're asking about how a human population or a wildlife population is going to respond exposed to a chemical, you cannot predict what's going to go on in that population. Like the mixture study, you're basically going to be able to predict a mechanism, which is why model species are powerful. In contrast, sentinel species are, in fact, the world that allows us to understand environment by gene or environment by physiology responses. These are real populations. These are populations like you and me. There's genetic diversity. Now, they may not be great for some mechanistic studies, and studying alligators doesn't really allow you to do multi-generational studies, since they probably live as long or longer than we do. But the take-home is that it's important for us to embrace all approaches, not just one approach. But of course, in any work, interpretation's critical. So as these two miners are trying to figure out, that dead canary does mean something. And the fact is, for basically 50 years, we've known that the environment isn't healthy. Rachel Carson told us that, but the fact was is she already knew that, okay? And folks around her knew that. But how do we interpret the world around us, and how do we translate that back not only to meaningful change, but how do we translate that back to talking about ourselves? And so it's the Lake Apopka story. 1985, my wife and I, Buzzy, who's in the audience and will be speaking in a little while, who's a professor in anthropology at Florida when I was there in biology, basically, I got a knock on the door, and there was a guy in a uniform there. I thought, oh my God, what have I done now? And he said, we hear you're a reptile expert. And so, well, I know a little bit about reptiles. I worked on high-altitude pregnancy. My career has always been how does the environment influence the development and functioning of the reproductive system. But see, toxicology, for me, when I grew up in the world, toxicology was bad science. You basically threw things at animals and killed them. And I wasn't interested in killing things. I wanted to understand how the environment influenced animals. So we show up at Florida, not many mountains in Florida. As you all know, I think the natural high spot's like 75 feet or something. But the take home is that the guy says to me, Woody Woodward from Fish and Game, we hear you're a reptile expert. We got this endangered species, the American alligator, and we want you to work out the reproductive biology of the alligator. And I started laughing, and I said to him, you realize there's 100,000 people today trying to figure out the reproductive biology of humans. What in the world are we going to do? Well, we surprised ourselves. I think we've learned a lot. And so what we started to do was to study the alligators, not just at Lake Apopka, but at 7 different lakes in the state of Florida. It was an endangered species, but yet it was a species that they wanted to actually use for agricultural purposes. There are farms. There are 50 farms. There are still people who actually like alligator shoes and alligator luggage. And so the take-home was, could we actually give value to these animals other than just to a bunch of, as we were called one time in a court of law, pointed-headed, greeny intellectuals? So was there something else that we could actually do? And so Woody and I set up what we called the innies and the outies. So they were the outies. So they took the skin out. My team took the skin in. And so what we would do is go out on warm summer nights. I mean, can you imagine this? You're a professor, you're getting paid, and you get to run around on an airboat at night, okay? And you basically would take grad students and hang them off the front of the airboat, okay? And those grad students had to make a snap decision, okay? If the animal was 5 feet or smaller, you grabbed it with your hands. If it was over 5 feet, you're supposed to noose it. If you made a mistake, then your dissertation took twice as long to type, okay? So it ends up that we did have a number of students that appeared at the infirmary with animal bites, and then the nurse would say, but that's not a dog bite. And they said, I didn't say it was a dog bite. I said it was an animal bite. But the take home is that we actually started to learn a lot of things. And one of the things we actually learned was literally how to work with alligators. Before we started, basically, if you wanted to study an alligator, you went out with a bang stick and you shot it, it killed it, okay? And we said, no, that's not our goal. This is an endangered species. How do we catch them? How do we actually get a blood sample? How do we get a urine sample? How do we do tissue biopsies? How do we do all these things that a vet does? And so I called a couple of zoos and they said, oh, no, we don't do that. We just put them in a tank and let them grow. And so I said, yeah, but, and they said, nope. So we actually spent a lot of years. I'm not pithing that animal. We basically found a blood vessel that comes out of the back of the skull and runs down the top of the spine. We published papers on how to do this. We teach vets now how to do this at zoos and aquariums all around the world. So we take blood samples. I went to my local vet and I said to him, show me every catheter you have. And so there were like 22 of them. We laid them out on the table. So I said, I want one of those and one of those and one of those and one of those. And he said, what are you doing? I said, well, I'm going to go catheterize alligators. And his response was, can I go with you? And I said, well, sure, come on, let's do it. And so, oh, I'm sorry, I'm too busy. But take home is, we developed all these techniques so we could actually try to understand this animal, okay, at a level to really get some sense of mechanistically how is it responding to the environment. So you go out on these warm, beautiful summer nights, or winter nights, or whatever night, you catch the alligators, you pull them on board with you. Our goal is basically to get samples from them, measure them, mark them, whatever, and release them, and do all of that in 10 to 15 minutes. And we can do that with animals up to about 10, 10.5 feet. Anything over that, you're now dealing with animals that are 800 or 900 pounds, and it becomes a little more exciting. But we do that too. The take home is what we started to realize when we actually started doing the innies and the outies was that the animals were not, in fact, the populations were not growing at the level they should. We were not getting normal recruitment. We weren't getting the numbers of babies we expected when we actually looked at numbers of nests and did nightlight counts and did all these sophisticated ecological models combined with all my genetics and endocrinology. It just didn't all add up. So we started to say, okay, well, if we have poor recruitment, that is poor reproduction, well, you have to go to the unit of reproduction. And so again, I'm very proud of convincing the National Institutes of Health to pay for helicopter time. We fly around. Why? Well, as some of you actually probably saw yesterday at Lake Apopka or anywhere else, these lakes are not the sandy beaches everyone thinks of in Florida. In fact, the guy who's standing on the seat of my airboat there is a 6'7" former offensive lineman for the New England Patriots who was one of my PhD students, and you can actually see that the grass is about level with his head. So that's about 12 or 14 feet deep. You can't see the nests, not easily, and so what you have to do is you put the helicopter in the air, you spot the nest. Just show you here. So here's the nest. Actually, Mama's sitting right here. Airboat comes in. Here's the nest with the guard pool. Here's, in fact, where Mama goes back and forth to the lake. That's where you send your grad students in, okay? Especially new ones. It's basically, students are a renewable resource at a university, so you send them in there. Mama will, in fact, defend the nest, and so many times we can catch her at the nest. We can take blood samples, et cetera, and then you open up the nest. Just a little basic information. Takes about 12 to 15 years to reach sexual maturation as an alligator. Females do it at about 6.5 feet. They will start breeding then. Males will not. Males literally need to get to 9.5 or 10 feet before they can compete with the big boys on the lake. The female will lay about 45 eggs, anywhere from 30 to 50 or 55, but let's say about 40, 45 eggs about every other year. We find on the coastal regions it's every third year. We find, in fact, in the inland areas, it's about every other year. So we go out, we collect the eggs. We also learned early on that if we went in and only took 4 or 5 eggs to measure for contaminants and stuff like that, that raccoons or wild pigs would actually follow us in the next day and take all the eggs. So what we, in fact, do is we collect all the eggs, we bring them to the lab, we incubate them, and we basically look at hatch rates. And that's been an incredibly powerful statistic for us. Just so that you know, in the last 25 years, we've actually done almost 400,000 eggs. So we have a pretty good data set. And I can tell you that the hatch rates in Florida are only about 50%. That's removed from predation, that's removed from flooding, et cetera. Should be 90%. So there are problems with the hatch rates. And what we do is, just so you know, is we take a very few number. We have a very small number that we actually take for research. All of those eggs, we know exactly what nest they came from, and after they hatch, we take them back and give them back to Mom, and Mom takes them back. And in fact, you can go back out months later or weeks later. Mama will actually stay with her young for about a year to 18 months. So it's much more sophisticated behavior than just these cold-blooded reptile killers that we all know about alligators. So, hatching, poor hatching. Lake Apopka in the early 1990s had hatch rates of 10%. 90% of the eggs were dying. And we actually spent some time thinking about mortality. But let me give you my logic of why I switched. Instead of actually spending all our time trying to figure out what killed them, what I wanted to know is, would the ones that hatch actually survive and be healthy adults? So here's my logic. We now know from mark and recapture studies, which we've done, that we have actually caught females 30 years after we caught them 30 years ago, and they're the same size and they're laying the same number of eggs. So we have a 30-year reproductive lifetime. Let's say that she only breeds every other year. That's 15 clutches. She's laid 45 eggs. That's a lot of eggs. How many offspring does she need to produce to reach sexual maturation to have a stable population? Right? Zero population growth. All you need to do is replace you and your partner, and the population's stable. So here is my question. is that little guy going to turn into a healthy adult? Because if so, instead of feeding raccoons and osprey, et cetera, baby alligators, we're just feeding raccoons eggs, okay? If in fact we're still getting 10% or 15% or 20% of the eggs coming out, will they in fact be healthy? Now, we don't think about that like humans, okay? It would be, in humans, mortality is a big deal. But we're thinking populations here. We're thinking about wildlife. So it's a slightly different spin. And the question was, would they be healthy? And so we started studying lakes. This is just one of my recent slides because we also now have 3 sites in South Carolina that we study. But we've been studying Lake Apopka for a very long time. And we actually studied Lake Woodruff National Wildlife Refuge. Basically, it's only 80 kilometers away. It is actually a beautiful place, never had modern agriculture around it, never had modern municipalities dumping stormwater or sewage or whatever into it. And that's been our 2 comparisons. Now, we study 7 other lakes. I'm not going to go into them tonight. But the take-home is that Lake Apopka was our worst case scenario. But let me put this in context for a minute. A couple years ago, I'm giving a talk in Sweden at the Karolinska Institute, and I'm doing this talk and I'm showing this stuff, and all of a sudden in the back of the audience, there are these guys that just will not shut up. They are just freaking out. And finally I said, okay, wait a minute, sorry. What did I just say that insulted you guys? And they said, no, doc, just ignore us. And I said, no, what's going on? So, well, we're a little worried because your Lake Apopka is what we're trying to restore the Baltic to, and you're calling it your highly contaminated site. So the fact is, Lake Apopka, I'm not belittling it, Lake Apopka is contaminated. But there's lots of other places in the world that are Lake Apopkas, I think is the point I'm trying to make, okay? So here is, in fact, a summary slide of 25 years of research. And the take-home is the following. Before that baby alligator is ever born, as still an embryo, or after it's born, we can already show that there's abnormalities in the anatomy, the physiology, and the genetics. Those abnormalities persist into juvenilehood, and we now have data showing that it actually persists into adulthood, okay? This is not baby fat that this animal is going to grow out of. These are, in fact, real phenomenon. And let me just today talk about just one example. So when we started this work, we tried to ask what was the fact, what was going on? Understand that I'm trained as a reproductive endocrinologist, developmental geneticist, comparative endocrinologist. I work on the evolution and development of reproductive systems. I'm not a toxicologist. I still don't believe I'm a toxicologist. What I'm interested in is environmental health. And so one of the take-homes we asked was, well, what's causing these problems that we're seeing? What's causing all of these weird things that we're seeing? The fact is that we didn't think about chemicals, believe it or not. It's really funny. Jay said he was down here, literally 5 years before I actually moved to Florida in 1985, dealing with Lake Apopka and all these issues here. I was 2 hours away in Gainesville. Lake Apopka was unknown. It was just someplace down near Mickey Mouse World, okay? And so the fact is the state picked Lake Apopka as one of the study lakes because they thought the population here was pretty reasonable as far as alligators were concerned. The take-home is it wasn't until literally 4 years into our study that we realized Lake Apopka had a Superfund site down by Gordon Neck Springs, which is on the southern end, and that is probably a Superfund site because of all of what was going on in the farming on the north end. But the take-home was we didn't know that. We started this study not because it was toxicology. So the take-home, we wondered, is it population genetics? We did sophisticated pop gen studies. We thought, well, maybe it's in fact what they're eating. That is, the kinds of fish they were eating because maybe the lakes have changed. Nope, that didn't explain it. We thought maybe it was the gas environment of the nest because we know that basically CO2 and oxygen are so critical. No, it wasn't that. We thought, well, maybe it's the temperature of the nest. No, it wasn't that. We finally realized it's not what they were eating, but it was what was in what they were eating. And what we in fact came to understand is that Lake Apopka had a fair amount of contaminants. Now let me just really, really briefly, and I'm trying to get through this so that we're still on time, but the take home is the following, is that Lake Apopka, and this is about the time we did this work, this was actually published, this initial paper in '99, okay? What we in fact found was we found all these contaminants. It's just a stacked bar graph, okay? Now, 2 things that are important. This is not fat levels. These are serum levels. Why is that important? Because what's stored in your fat, stored in your fat. But what's circulating around in your blood is what your cells are exposed to every day. That's why the NIH and our group as well basically have switched what we're doing. We basically look at what's circulating in the body because that's what you're exposed to. But let me just make a really important point. Orthoparaprime DDT, we all know DDT. Did you know that in 1952, DDT was actually shown to be estrogenic? Series of studies published at Cornell Cornell Ag School showed that DDT was an estrogen. Of course, 40 years later, I'm arguing that DDT is an estrogen. They're telling me, no, there's no evidence for that. Okay, that's 40 years old. DDD, one of the major metabolites of DDT, it's been sold as a drug in this country for 50 years under the trade name Midotane. It was, in fact, the original drug that we used to treat adrenal cancer. Why? Because it blocks adrenal steroidogenesis. We still use it, some in the vet world, but we have fancier drugs now. We're arguing there's no endocrine disruptors. We're selling a metabolite of DDT as a drug to disrupt the endocrine system. And of course, DDE, we now know the major metabolite is an anti-androgen. It actually blocks testosterone functioning and androgen functioning at the receptor. So we have a wide array of chemicals. We know that there's lots of other chemicals. The fact is there are phthalates in BPA. Actually, the highest phthalates we've recorded in alligators in the papers just being submitted is actually down in the Everglades. It's actually not on Lake Apopka in Central Florida, but there are detectable levels. In fact, they're much higher than we actually see in humans, believe it or not. And of course, there's atrazine, as you'll hear later today, and a wide array of other chemicals. I put this data up really quickly because we've spent so much effort on Lake Apopka. This is 2010 data. And I just want to show one thing here. This is DDT. This is Lake Apopka. Note the scale change. It's an order of magnitude different than all the other lakes that we study. Okay? The fact is, this is 2010. We have data coming out on eggs that we're just finishing the analysis on. We've measured 250 organochlorines in alligator eggs. And what we in fact find is that the DDTs, toxaphene, transnonochlorodanes are all extremely elevated in Lake Apopka compared to other sites. The levels are not different than those were measured in 1985, okay? It's not changed. So the old advertising for DDT, Duradust, its action lasts and lasts and lasts. I don't know if you saw those ads. They were correct, okay? The other important point, take home really briefly, is that we measure what we can measure. You go to a restaurant, you lose your keys. What do? You walk back to your car and you look under all the street lights looking for your keys. Why? Because that's where the light is. We are still studying and measuring what we can study and measure, what we think is important. But when I do a mass spec analysis of what's in alligator eggs, I get 400, 500, 600 peaks. I can only identify 100 of those. So there are hundreds of chemicals which are present, that some natural, but many of them in fact not, that we can't identify yet and have never been identified in many cases, and we have no idea what the health consequences are. So just because you have contaminants, just because you actually have problems, does not mean that you have causation. And so what we've actually spent a lot of times looking at alligator eggs, looking at their ovaries. One of the major pathologies we found in the alligator, I'm not talking penises today, okay? I'm going to let the guys relax, no crossing legs, no freaking out. We're going to talk, we'll pick on the women for just a few minutes today. There's an ovarian abnormality here. This is a multiocytic follicle. A normal follicle should only have one egg and one nucleus. What we in fact hear is multiple eggs, multiple nuclei in an ovarian follicle. This is a pathology called the multiocytic follicle, polyovular follicle. This was originally described in little girls and in rodents exposed to diethylstilbestrol, a pharmaceutical estrogen. This was in fact what Howard Byrne and Tayaguchi discovered a number of years ago. And the only cause that we know of this is abnormal estrogen signaling during embryonic development in a female. This is associated with infertility. It's associated with early embryonic loss if you can even ovulate these eggs. Question is, where were my alligators getting estrogens from because they were not getting a pharmaceutical estrogen from their doctor? Now, why estrogens are important? Well, it ends up that one of the most critical of this whole estrogen signaling is an enzyme called aromatase. It converts testosterone to estradiol. The reason men do really stupid things on a regular basis is because we don't have much aromatase, okay? So women convert most of the testosterone that they make into estrogens. That's why they use most of their brains. That's why they do really smart things compared to the rest of us. But in order to have estrogens, you have to have an estrogen receptor. And early on in the alligator, remember, these are early days. This is 20 years ago. The take-home was we didn't even know if alligators had normal estrogen receptors. We now know they have a normal estrogen receptor, just like we find. They, in fact, have two. It binds to the DNA like it's supposed to. One of the most stunning things we found is that the DNA binding region of the estrogen receptor in the alligator is literally 100% conserved with that of humans. The fact is there is no variation in the DNA binding region of the estrogen receptor between alligators and humans. It tells you how important the estrogen receptor is because just a slight mutation and you are not represented in the next generation, okay? You have to have functional estrogen receptors. The the hormone binding region is 95% conserved. It's found on chromosome number 1 in alligators, just like it is in us, in birds and other species. We've been able to take those receptors, basically make molecular machines. We can now take the receptor from 50 different species that Tayaguchi and our labs have cloned out over the last few years. You can do tests. We test chemicals. We test hormones with it. You actually take the gene, you insert it in a plasmid, you put it in the cell. You take any chemical you want to test to determine if it's an estrogen or an anti-estrogen, an androgen, an anti-androgen, whatever. You put it in, it reads out. And those graphs just say alligator receptors are perfectly good, and they work at 10 to the minus 12th. We're talking about part per trillion or lower concentrations. The other thing is we've actually shown that chemicals like transnonachlor actually bind the receptor. They take a bit more, but the fact is they're actually giving us almost 100% gene expression. So the take home is that transnonachlor is a perfectly good estrogen, like lots of other things. So why study alligators? She has no ovarian follicles when she's born. She is still growing her follicles. In contrast, she has every ovarian follicle she's ever going to have at 7 months in utero. We can't study, well, besides the ethics issues of studying little girls and little boys, we can't study that because it's in utero. But the alligator we can. And so what we've actually done is challenge these baby alligators with the same kinds of stuff we do in a clinic, for example, with women. We challenge them with the drug, follicle stimulating hormone, that turns on the ovary. And what we in fact found was that, sure enough, the girls, in this case, girl alligators, exposed to this drug would not only increase the production of estrogen, like they're supposed to, but if they're exposed to agricultural chemicals from Lake Apopka, they also make more testosterone. That's not normal. And so we started to ask, what's the difference? Well, without going into a huge kind of story, we're way down into trying to understand mechanisms here. We now know how genes are regulated, which we didn't even know a few years ago. In promoters of genes, there's little regions that you can tag, and you can either turn on or shut off that gene. And it ends up the aromatase gene, that gene that makes estrogens, has 6 little sites in its promoter. And if you put methyl groups, forget what that is, you just stick things on those 6 little sites, okay? You can't turn on aromatase, which means you don't actually have the enzyme, which means you can't make estrogens. That's what boys do, okay? That's what these blue bars mean. They actually have lots of those stickies in the promoter. In contrast, girls have less. Why? Because they need aromatase. They make estrogen. What's the take-home message of this whole talk? It's this slide. If you look at the females from Lake Apopka, the day they're born, those females' ovaries are what we call hypermethylated. They are already halfway between what should be a normal girl and a boy. They already have suppressed aromatase activity. They cannot make estrogens like they're supposed to. That's why when you challenge them, they make some estrogens, but they also make androgens. What's that like? PCOS. Polycystic ovary syndrome, which is epidemic in this country, that's one of the major symptoms of PCOS. Now, where are we? We're in the middle of we've actually gotten 50 women who come to an infertility clinic, have volunteered. They've given us samples. We now actually have granulosa cells from their ovaries, from their follicles. We have their follicular fluid. And we're in the middle of actually doing exactly the same thing now with humans. Why are we doing this in humans? Because the alligator told us that's where we should, in fact, be looking. So, gonadal dysgenesis is not just Lake Apopka alligators. We now have data from every vertebrate group from all around the world telling us that gonadal dysgenesis is real. We also know that we are not just a product of the genome that we got from mom and dad. We are a product of the environment that we grew up in. And finally, I hope I've shown you today that we can't just do applied science. If you don't understand basic science, you can't do it. How do I know this cartoon was drawn by a guy? He has no idea what the udder is for, okay? You have a cow with a second set of mammary glands. And certainly never took general biology from me because you got a bird with mammary glands also. The take home is that there are clearly species differences. But what's also important is that if I give an estrogen to a baby girl alligator and a baby girl, I get the same ovarian pathologies. At the ovary, at the genetic and molecular level, an ovary is an ovary. A testis is a testis. And so they are telling us something about our own lives. I didn't tell them I was going to do Rachel Carson, but here's Rachel Carson. It's ironic to think that man might determine his own future by something so seemingly trivial as the choice of an insect spray. Rachel Carson, Silent Spring, 1962. No gene theory, no epigenetic theory, no modern endocrinology, no knowledge of endocrine receptors, but the woman was brilliant. She was a brilliant observer. And so I add my own little comment. Well, how about personal care products? How about drinking cups? How about the many other things that we're exposed to today? So I'm basically standing up here representing a whole bunch of people. As you know, this kind of complexity is no way in the world can one person do it anymore. Modern science is a partnership. The guy up in the corner, Woody Woodward, is the guy who got me into hot water playing with alligators in 1986. Howard Byrne, I've already talked about. This is my current group at South Carolina. It all takes money. There never seems to be enough. There's never enough time, but we try. So what we try and do is to try and figure out how she generates a healthy one of those. The fact is, if the environment's not healthy for her kids, I don't think it's healthy for ours. Is it a one-to-one relationship? Of course not. But if they're basically telling us the environment is dangerous for her kids, then we have to, in fact, be aware of what the world is to our kids. Thank you very much.